Okay, so I'm going to talk a little bit about bipolar disorder and the fact that it runs in families. And the fact that it runs in families is a reflection of the idea that there's genetic risk for this illness. So William Styron was a famous author of American literature, and he had a wonderful expression of what it was like for him to suffer repeated episodes of depression. It's really wonderfully elegant prose. And Theodore Roethke was a famous poet, American poet, who had bipolar disorder as well, and this was his description of what it was like for him in a brief period of time during one of his manic episodes. To give you some flavor of the richness of this illness and the uniqueness of the experience of people with it. So a couple of principles of psychiatric genetics, and here's a take-home message. All of the DNA variations that increase risk for these disorders that are common in our population increase risk individually by a tiny amount. So there's no reason for people with one of these illnesses, be it schizophrenia, bipolar disorder, recurring depression, OCD, etc., to have their DNA sequenced by 23andMe or some other company with the idea that that's going to help them understand the illness. That's just not true, sad to say. Nor is there any reason for family members who might be at risk for the illness to have their DNA sequenced. This is not going to tell them anything about their risk for illness, unfortunately. Not at the present time, although in the future that might be possible. So the situation in psychiatric illnesses are that these common DNA variations some of them are in every one of us, so that there may be hundreds to thousands of DNA variations which increase risk for schizophrenia. Similarly, hundreds to thousands that increase risk for bipolar illness, but each one does so in a tiny way, so that people who develop these illnesses have to encounter the right environmental events, and they typically have to inherit dozens and dozens of these risk-increasing DNA variations, some from mom and some from dad, so now you can blame both your parents. One of the interesting aspects of the genetics is we used to think of bipolar disorder and schizophrenia as completely separate illnesses. But in reality, they share quite a few clinical characteristics as shown here. And one of the most amazing discoveries of research of genetic research in this area is that bipolar illness and schizophrenia share a lot of genetic risk. And this was a complete surprise to us. So you can see here that people with bipolar illness and people with schizophrenia have higher risk for suicide. They both can have hallucinations and delusions. They both can have cognitive deficits. There's increased risk for drug and alcohol addiction in both types of illnesses. Interestingly, both can be treated by atypical antipsychotic medicines. And both kinds of disorders have an elevated risk for recurring depression in their relatives. So here's one example of a rare allele that has a large influence on risk. And in this rare allele, there's a break, an abnormal break in one of our chromosomes, chromosome 1, and it got attached somehow to the end of chromosome 11. This is called a translocation. And here's a famous kindred or family in, that has this translocation. You may not be able to see it in the back, but there's a little asterisk here that tells who in the family has this translocation. And when this symbol, which represents either a man or a woman who's a child of these two people, when this symbol is completely filled in, the individual has a diagnosis of schizophrenia. When it's half filled in like this, it's depression, and or what I like to call recurrent unipolar illness, RUP, in this slide. So there are 10 people with this translocation who have depression, and 7 that have schizophrenia. So even when we have this terrible genetic event that has a large increase in risk, the end result can be either schizophrenia or depression. And we don't understand why. So there are lots of these rare, large, unfortunate genetic events that predispose to schizophrenia, and sometimes the end result is depression or bipolar disorder. But one of the things that we've been able to do with modern DNA technology is to do a variation of sequencing the entire genome, all 3 billion base pairs of DNA, in lots of people to try to find the common variation that predisposes to schizophrenia and bipolar illness. And this has been done with thousands of patients who carry these diagnoses, and in thousands of people who never had such an illness. And the reason we can do this is that this common kind of variation called the single nucleotide polymorphism, or SNP, is very automatable. That is, we can do it very rapidly and large scale. And so we use one of these SNP chips, as it's shown here. So simply incubating the person's DNA with this SNP chip and putting the chip in a machine gives us the DNA sequence of that person, and we can align these sequences from thousands of people with bipolar illness and thousands of people without that illness and look for differences. And that's essentially what we've done. And you can express this kind of information in what's been called a Manhattan plot, because there are these so-called tall buildings. So every little dot here represents one DNA sequence variation that's been tested to see if it's more common in people with bipolar illness compared to people without that illness. And here you can see that this is the name of one gene that seems to have sequence variation in it that predisposes to bipolar illness. And this gene was known to geneticists before this study was ever done, because it causes different mutations in it, cause a rare syndrome called Timothy syndrome, which is autism-like features and irregular heartbeat. So this gives us pause, because the same gene, different variations in it, can produce markedly different clinical appearances, both brain disorders. Curiously enough, the protein that this gene produces binds a medicine that's FDA approved for the treatment of bipolar disorder, and that's called lamotrigine or Lamictal. It gives us some idea that maybe we're on the right track here, but probably not. So the same kind of thing was done for patients with schizophrenia, and here you can see that they 36,000 participants with schizophrenia and 105,000 people without schizophrenia went through this same process. And so one of the discoveries was this, some of the same sequence variations that increase risk for bipolar illness were found in this study, but lots of other ones too. So using this kind of technology, we hope to get to better treatments by understanding the genetic risk. That's the sole reason we're trying to do this, is to get to better treatment. The same thing has been done for major depressive disorder or recurrent unipolar illness. And here, in this study of 5,000 individuals with major depressive disorder and 5,000 control people, all of Chinese ancestry, they found sequence variations in 2 genes, SIR2 and LHPP. And I'm saying LHPP because I can't pronounce the real name of the gene, which is this. And you shouldn't try to pronounce it either. Unfortunately, none of these 2 genes gives us an immediate handle on developing treatment. That is, we don't have known medicines that interact with either of these 2 genes. And so new ones are going to have to be developed. So much to our surprise, after this was done, we began to compare the results for bipolar illness and schizophrenia and major depressive disorder. And lo and behold, we found that the risk alleles in each of these illnesses were shared across illnesses to a much greater extent than we predicted. So here you can see in the light green columns the amount of risk alleles that are shared between pairs of disorders. So right here is schizophrenia and bipolar disorder sharing, bipolar disorder and major depressive sharing, schizophrenia and major depressive sharing. And this is highly significant because you get sharing of like 10 to 15% of the genetic risk across these illnesses. So it seems to us that how we're defining the illnesses has to get better and has to recognize this shared genetic risk. I'm kind of an old psychiatrist, and when I started in training, lithium was the only FDA-approved treatment for bipolar illness. And I still use it a lot because we have 50 years of experience with it. One of the embarrassing parts about this is that after using it for 50 years, we still don't know how it works. So there's progress for you. But I like to use it because of these quasi-beneficial aspects. And I was part of a large study conducted in 12 sites across North America and Europe, in which we tried to find people with bipolar disorder who were doing really well on lithium as the only medication. So these were people who might have had lots of episodes of depression and mania. They got finally onto lithium, and the medicine prevented the subsequent episodes. And so we looked for these patients in our medical records, etc. And so those patients were termed lithium responsive, or LR. And then we could also find, much more easily, patients who did terribly on lithium. You know, they took the medicine, but they continued to have episodes of depression and mania. So these were called lithium non-responsive. So we took some skin cells from these patients, and using some laboratory tricks, in tissue culture, we coaxed them to become neurons. Because after all, bipolar disorder is a brain disease, it's not a skin disease. But, you know, we really couldn't take brain biopsies, so we took little tiny pieces of skin, and using some interesting proteins, we could coax them into becoming nerve cells in a dish. Don't try that at home. And we were really quite fascinated by the results. And this is a busy slide. Ignore that stuff on the end. It's for genetic types. But here is the important point. So you see, we also did this in people without bipolar disorder, and each one of these spikes here represents a time when this particular neuron fired. Now, you don't have to be a doctor to see that the neurons from the people without bipolar illness are much quieter than the neurons from the people with bipolar illness. And these just are the statistical analyses which show that. So there's something about these neurons that make them hyperexcitable, and we don't know what it is. More interestingly, if we separated them out into the people who are lithium responsive and not lithium responsive, and added lithium to the tissue culture, the people who are lithium responsive, their neurons got quiet. The people who are lithium responsive, as shown up there, their neurons didn't change that much. They were still hyperexcitable. And this is like the statistical analysis here. You can see it pretty well here. Here's the lithium responsive people. Here's the excitability of the neuron. You add lithium in, and the neuron becomes quiet. You don't see anything like that in the lithium non-responsive people. So here we have a $100 million test to see who's going to do well on lithium. And it's our job to make it into a $100 test sometime in the next couple of years. Maybe actually sometime in the next decade, actually. This is an important kind of advance, potentially, because all psychiatrists need this kind of information. They need some sort of test that says, my patient sitting in front of me is going to do well on lithium, so I'm not going to prescribe anything else except lithium, and we'll see what happens. This kind of pharmacology advance may help people with bipolar illness. And given the sharing in genetic risk between schizophrenia and bipolar illness, we might also identify some people with schizophrenia who would do really well on lithium. And so that's the remarks I have, and at the end of the day, I'll be glad to answer questions. Thank you.